This year's NYU Shanghai iGEM team, represented by Ziyu Zhao, Zhongling Liang, and Sirus Mrazik, aims to introduce a new biological way in the alcohol production industry to consume the methanol in fake alcohol. Fake alcohol production in China has increased dramatically over the past few years as people continue to seek large profit from the increasing popularity of the bar industry. Fake alcohol can be considered as either illegally made drinks produced at home or drinks made with cheaper alternatives that are placed in brand bottles and passed off as being the real thing. We decided to focus on one of the most commonly added replacements, methanol, which is distinct from the real alcohol, ethanol, because it proves to be very dangerous, causing blindness or even death. At present, chemical and physical methods are more commonly used in the industry to decrease methanol, but due to the high similarity of methanol and ethanol, ethanol is often reduced as well. Therefore, a more efficient and effective method is necessary. Here we propose the biological solution to this problem, since enzymes are very specific to their substrates and will not interact with other molecules. We were inspired by the Methylobacterium, a group of the oldest bacteria in the world. While methanol is one of their energy sources, they cannot metabolize ethanol without the help of other bacteria. This is precisely the type of organism that we were looking for, one that can metabolize methanol without affecting ethanol. Recently, scientists have successfully figured out its unique biological pathway, which converts methanol to methane with the help of enzymes. From these studies, we found out the essential components in this conversion, the enzyme MTAB and the coronoid protein MTAC. Since methane is a greenhouse gas, we decided to stop the biological pathway halfway at its intermediate methyl MTAC. Since the Methylobacterium are highly difficult to grow, we chose E. coli as our chassis and decided to transform the MTAB and MTAC genes into E. coli cells. E. coli is most commonly used in bacteria in genetic engineering experiments, and there are many compatible plasmids for E. coli. We finally chose PET-duet1 as our carrier plasmid because it has a capacity for 2 genes as well as the gene for ampicillin resistance, which is a selective marker for successful transformation. The summary of our project design is simple, but of course all things are easier said than done. We have run into many hurdles along the way which have truly pushed us to become resourceful and dig into our research. In considering our experimental model, there were important questions to consider before we proceed to enzyme testing. Can the E. coli survive long enough in methanol and ethanol to finish their job? What is the best way to detect the methanol concentration to check our hypothesis? We have tried many methods to answer these 2 questions and finally attain reliable results. We tried to answer our first question with 3 methods: spectrophotometry, fluorescence microscopy, and clonogenic assay. Our first method, spectrophotometry, was designed based on the fact that when a bacterium dies, its membrane breaks and all the proteins are released. The spectrophotometer is a machine that can be used to detect the concentration of these proteins by measuring the light absorbance of a certain wavelength. By measuring the change in protein concentration after adding methanol, we sought to find the cell death rate according to the formula found in the literature. But when actually put into practice, the reading fluctuated considerably and so was unable to give us an accurate result, probably because of the protein settling and degradation in methanol. So we decided to find alternative methods to determine the E. coli survival rate. The second method we tried is double staining fluorescence microscopy. Like the first method, it relies on the fact that bacterial membranes lyse upon death. 2 fluorescent dyes, propidium iodide and Hoechst 33342, both stain bacterial DNA, but they differ in their permeability and in their fluorescence. Hoechst 33342 is a nucleic acid stain that emits blue fluorescence when bound to double-stranded DNA. This dye is highly permeable and can enter the cell membranes of both dead and live E. coli. This dye serves as our counterstain and control, allowing us to visualize the location of all the E. coli under the microscope. The other important fluorescent dye we used is propidium iodide, which emits red light when bound to DNA. Unlike Hoechst, propidium iodide can only penetrate the cell membrane when E. coli is undergoing lysis, indicating that they are dead or starting to die, making it a very useful tool to test the survival rate. We had to play around with different conditions to see which ones will give us the best result. For example, we tested different dilutions for the bacterial suspension, different concentrations of the dyes, incubation time, dye ratio, and amount. We also had difficulties at first finding our E. coli. There are a lot of residues on both sides of the cover slips that we accidentally mistaken as E. coli. After deep cleaning our microscope slides and cover slips with the ultrasonic cleaner, we finally identified our E. coli under 400 times magnification. After numerous testings, we found that 50% methanol is deadly to the E. coli, as indicated by all the red fluorescent signals. We overlapped the blue and red images to visualize how much E. coli has died. The purple in this figure denote bacteria which are dead, while the blues show those which are still alive. As seen here, there is a large amount of purple with very little blue, thus showing those cells which are about to die or already lysed. Additionally, we determined that ethanol had a more detrimental effect on the E. coli, with 30% being the fatal concentration, showing similar results here. In order to determine whether or not our fluorescence microscope results were reliable and accurate, we utilized another method called clonogenic assay to test the E. coli survival rate in ethanol and methanol solutions. Clonogenic assay is a technique commonly used in cancer research to study the effectiveness of specific agents on the proliferation and survival of cells. This method involves applying a treatment to a sample of cells and then spreading these cells on agar plates to allow them to grow overnight. The next day, you can observe distinct colonies of bacteria on the agar plates. The number of colonies able to grow on the plate signified the number of live bacteria originally deposited onto the plate. Comparing the number of colonies on the treated plate to an untreated plate gives a rough idea of the percentage of cells that survived the treatment. For our experiment, we first determined the ideal dilution of our bacterial suspension for a visible result. Without a high enough dilution, it will result in a bacterial lawn rather than in distinct colonies. After diluting our overnight bacterial suspension by 10,000 times, we were able to obtain fairly decent results. As for the treatment, we tested various concentrations of ethanol and methanol, ranging from 10% to 50% with varying incubation times. After incubating the E. coli with the treatment for a certain period of time, we removed the methanol-ethanol solution, diluted the sample using LB nutrient broth, and spread 10 microliters of that sample on the agar plates. As we increased the concentration of ethanol and methanol, we observed a slight decrease in the number of colonies and the colony size. In the figure shown here of our methanol-treated E. coli, the number of colonies significantly decrease in size at 40% and do not even grow at 50%. Similarly, in the ethanol treatment test, there appears to be very little change between the control and the 20% ethanol treatment, but no growth at all under the 30% treatment. These results, that no colonies form at 30% ethanol nor 50% methanol, are consistent with the results we found using the fluorescence microscope technique. After determining the maximum concentration of ethanol and methanol that the E. coli can survive in, we moved on to conduct testing on our genes. The first thing we did was make our E. coli take in the plasmid containing our two genes via transformation, more specifically the method of heat shock. We ordered competent DH5-alpha strain of E. coli. The E. coli were treated with calcium chloride solution to increase the ability of the E. coli to uptake the plasmid DNA, thereby making them competent. The plasmid can enter the E. coli upon heat shock, which includes chilling the cells on ice for 30 minutes, placing the cells in a 42 degrees water bath for 50 seconds, and immediately back onto the ice for 2 minutes. After transformation, we spread these cells on LB ampicillin agar plates. Only cells that took in the plasmid, which contains the ampicillin-resistant genes, can now survive on the plate. After the bacteria has successfully transformed, we need to test the feasibility and efficiency of our pathway by monitoring the change of methanol concentration at specific time points after transforming the E. coli. This leads us to our second question: how do we measure the methanol concentration? At first, we proposed to use gas chromatography. This precise method is commonly implemented in organic chemistry to separate easily vaporized organic compounds and determine their relative amount. For our initial test, we planned to use a simple solution of methanol and water to test the pathway's effectiveness. If the pathway does indeed work out nicely, then we would proceed to test solutions more similar to real and fake alcohol in order to further expand on application. However, the gas chromatography machine in our lab cannot test water solutions because the water in the solution will damage the machine due to its polar nature. we considered methods to extract the methanol from the water or to use a solvent other than water, but it is very difficult and more trouble than it is worth. Thus, we moved on to an alternative method, magenta sulfide colorimetric, which is a commonly used chemical method to detect the methanol concentration in the alcohol industry. The basic mechanism consists of 3 different solutions. The first, potassium permanganate phosphoric acid solution, oxidizes methanol into an aldehyde. The second, oxalic acid sulfuric acid solution, removes manganese dioxide produced from oxidation. The third, aniline red sulfinic acid solution, reacts with the produced aldehyde to change the color of the solution. As a result, the absorbance at 590 nanometers will change as observed with a spectrophotometer. The first step is to build a standard curve to find a mathematical relationship between the absorbance at 590 nanometers and the methanol concentration. Solutions of known concentrations will be treated with the 3 solutions, and then the absorbance is measured. Then the transformed bacteria will be added into a methanol solution. Then, after certain time intervals, the 3 solutions will be used, and after the E. coli is removed, the solution will be diluted and the absorbance will be taken and plugged into the standard curve to get the methanol concentration. We expect to see a decrease in concentration over time. Because it took a while for a gene and chemicals to arrive, this step of the experiment has not yet been carried out, but it will be conducted in the next week of experiments. One of the key lessons we learned was of the importance of planning and thorough research. We truly understand most of the time is not spent on the lab work, rather it is spent on the research. In our experience, plan in advance. It's always easy to say, but in real experiments, different incidents happen every day, whether the companies miss the time to ship you the chemicals or some methods are not practical in your experiment. So if possible, plan ahead to leave yourself more time so you're not always in a hurry.